Hi there, back to 100% Canadian maple syrup. Host always and forever, Al Wooden, aka the Skeptical Canuck. And tonight we're going to go over some very interesting evidence. That's right, evidence, including white papers from actual university experiments, and if I'm not mistaken, a company or two, as well as a article or two from a reputable tech website that covers these kinds of issues on the regular, and also a couple of videos to show you exactly what it is we're talking about here. So anyways, without further ado, let's get started. Oh, but first, before we do, please be sure to smack like and go ahead and subscribe if you haven't already. We have a few different podcasts here on this channel where we have guests on. We talk about different stuff. As well as some more stuff coming up in the future, a lot of pot smoking related stuff. But if you're not into that, a lot of serious article, news, and politics type stuff as well. So let's get into it. Make sure the best thing you can do for the channel is go ahead and share the video. So let's bring up the share screen. There it is. And we're going to go ahead and start off with an article that I stumbled across while watching the Corvette Report at CorvetteReport.com. Could injectable microbots one day run in your veins? Scientists have created an army of microscopic four-legged bots too small to see with the naked eye that walk when stimulated by a laser and can be injected into the body through hypodermic needles, a study said Wednesday. Laparoscopic robots are seen as having an array of potential uses, particularly in medicine, and U.S. researchers said the new robots have potential to explore biological environments. Of course, they're referencing they're either referencing bodies here, or you'll find out later environmental type things. Ecosystems, rather. One of the main challenges in development of these cell-sized robots has been the combining control circuitry and moving parts in such a structure. The robots described in the journal Nature are less than 0.1 millimeter wide, and I'll show you a scale in a moment, around the width of a human hair, and have four legs that are powered by onboard solar cells. That's right, the legs move, people. By shooting a laser light directly into these solar cells, researchers were able to trigger the legs to move, causing the robot to walk around. And this might sound crazy, but I'll show you exactly how they do it in a few minutes. The study's co-author, Mark Miskin of the University of Pennsylvania, told AFP that a key innovation of the research was the legs. Its actuators could be controlled by using silicon electronics. 50 years of shrinking down electronics has led to some remarkably tiny technologies, he said. You can build sensors, computers, memory, all in a very small space, he said. But if you want a robot, you need actuators, parts that move, figuring out what's possible. The researchers acknowledge that their creations are currently slower than other microbots that swim, less easy to control than those guided by magnets, and do not sense their environment. The robots are prototypes that demonstrate the possibility of integrating electronics with the parts that help the device move around, Miskin said, adding they expect the technology to develop quickly. The next step is to build sophisticated circuitry. Can we build robotics that sense their environment and respond? How about tiny programmable machines? Can we make them able to run without human intervention? Miskin said he envisions biomedical uses for the robots or applications in material science, such as repairing materials at the microscale. But this is a very new idea, and we're still trying to figure out what's possible, he added. Swallow the surgeon. Let me just go down here a little bit. All right, swallow the surgeon. Researchers said that they were able to produce components for the robots in parallel, meaning they could work more than 1 million of them in a 4-inch silicon wafer. They could fit them. The legs were made of nanometer thick platinum that bends when stimulated by a laser light, creating the walking motion. Their average speed was about 1 length per minute. There's study said, adding this was comparable to crawling biological microorganisms. The robots can survive in highly acidic environments and temperature variations of more than 200 degrees Kelvin and negative 70 degrees Celsius. Oh, that's the same temperature. The study said. In a commentary also published in Nature, Alan Brooks and Michael Strano of the Massachusetts Institute of Technology said the concept of the robot small enough to travel through blood vessels has been around since the Nobel laureate Richard Feynman talked about the potential to swallow the surgeon in 1959. We'll also give you another quote from that in one of the white papers we're about to read. They said the new study provides a clear vision for solving the challenge of creating a tiny robot that can both convert energy into motion and is able to be programmable. The authors robots, although not autonomous in their current form, can be seen as a platform to which brains and a battery can be attached. They said, predicting the hurdle of developing autonomous programmability for micro robots will soon be overcome. And so let's go ahead and check out another article that's a little bit more threatening. This one's entitled, Could Injectable Micro Robots One Day Run Inside Your Blood? Well, let's read from another fine fellow, Mr. Ray Kurzweil, who says they will be in your blood by 2030. And notice how it's said. I'm not sure if we went over this yet, but it's going to mention, as of yet, they don't have batteries, but they do have things that can respond to stimulus. I'll show you in a few moments how not only do they not even need the stimulus, but they can actually engineer it directly into whatever animal or human being that they want. But first, let's go over this really quickly. This is from futurism.com. And it's entitled Kurzweil. By 2030, nanobots will flow throughout our bodies. This fleet of tiny robots would keep you healthy and connected to the cloud. Now, obviously, I'm a bit of a conspiracy theorist, but when it comes to this, if something can keep you healthy, could it not also make you sick? And when I explain a little bit about where this technology is today, yes, it could do almost anything they wanted to. So let's go ahead and check this out. Ray Kurzweil, Google's director of engineering, is a well-known futurist who seems to have a penchant for accurate predictions. Most recently, he has again reiterated his prediction that so-called technological singularity will happen by 2045. For Ray Kurzweil, this doesn't translate to an end of the world as we know it scenario, courtesy of an artificially intelligent machines. Rather, it means human beings will become powered by machines. Now, notice he didn't say will become empowered. He says, unless this is a typo, that human beings will be powered by machines. And in fact, what you're going to see over the course of the next few moments is they're trying to replicate very similar structures that already exist in nature. And I think since they're at such a small level, perhaps they're even trying to replace the immune system. I'm not sure. Kurzweil believes that as part of this human machine meddling, nanobots will inhabit our bodies by the 2030s. While flowing through our arteries, these microscopic robots would keep us healthy and transmit our brains into the cloud. Forget brain chips. Small microbots would transmit your brain into the cloud. So they're not even trying to hide it. They're not even trying to make it look wholesome in that manner. They're trying to make it seem like a good thing. So this article it's not very long. It mentions another futurist, Dave Evans, founder of CTO of Silicon Valley stealth startup Stringify, gave his thoughts about Kurzweil's nanobots idea interview in a James Besdell in February. And he explained that such a merging of technology and biology isn't all that far-fetched. In fact, he describes three stages as to how this will all occur. The wearable phase, where we are today, the embeddable phase, where we're headed with neural implants and such, the stuff that Elon Musk likes to talk about while dissecting pigs live on TV, vivisecting, and the replaceable phase. It says the replaceable phase. I didn't have time to look at what that meant. I read these incredibly hard to read academic papers, which I'm about to show you. Didn't have time to look that up, but it doesn't seem too wholesome. So, and they finish off by saying, does Evans agree with Kurzweil's ideal of nanobots flowing inside our bodies? Check out the rest of his answer in the video embedded here. We're not going to do that, but there is a link in the description right now that you can check that out. Hopefully, after the show, hopefully you stick around long enough to see what we're going to talk about next. Next, really quick, I've read this article before on the show. IEEE Spectrum .org. The next generation of non-neurosurgical neurotechnology N3 program will fund research on tech that can transmit high-fidelity signals between the brain and some external machine without requiring the user to be cut open for rewiring or implantation. The program was announced in March, and Imondi, one of the researchers working on it, if I'm not mistaken, currently is picking researchers who will be funded under the program. He tells IEEE Spectrum to expect an announcement in early 2019. This is from 2018. Let's see. Now, you can read this for yourself again, but I'm going to go ahead and bring up what I think is relevant for time's sake here. So the program has two tracks, one for researchers developing completely non-invasive tech and the other for those working on minutely invasive technologies. Let's see. So it goes on to say, it explains, let me see where it is. I just want to read this one definition for you guys. About, is it not here? Huh? Yes, it is right here. All right, well, screw it. I'll just read this highlighted section here for you. The N3 program demands non-invasive tech that can read signals out of and write information into 1 cubic millimeter of brain tissue and do this within 10 milliseconds. To get this spatial temporal resolution, despite the barrier of the skull, Imondi says researchers would have to find new ways to detect neural activity. When a neuron fires, it changes its reflectivity. Can we capture that optical signal? He says. He asks, when it fires, it actually emits an acoustic wave. Can we capture that? Minutely invasive. DARPA invented this wording to avoid the term minimally invasive, which in medicine typically means laparoscopic surgery. DARPA doesn't want its new brain tech to require even a tiny incision. Instead, minutely invasive tech might come into the body in the form of an injection, a pill, or even a nasal spray. Imondi imagines neurotransducers, which I'll show you in a moment, are not only not needed, they've already been engineered in a way that they can be administered via a virus. That's right, a bioengineered virus. Anyways, he goes on to say that he imagines these neurotransducers that can sit inside neurons, converting the electrical signal that naturally occurs when it fires into some other type of signal that can be picked up through the skull. This is a 4-year program, and by the end of it, researchers want their tech to have a defense-relevant task. That deadline is fast approaching. I wonder if that is around the same time for the gene therapy that is being pushed upon the public right now in the form of injections. I forgot to read this one part, the introduction. Let's, oh, maybe not. Okay, well, I guess it's on this next page right here. You'll see what I'm talking about. This is also from IEEEspectrum.com. The first time Karl Deseroth used light to control brain cells in a dish, people had a lot of questions. 3 in particular. Can the technique be used in a living animal? Can it target different types of cells? Can it work without implanting a light source directly into the brain? In the years since that initial groundbreaking 2004 experiment, Deseroth's team and founders have found the answer to the first two questions, yes and yes. This month, they answered the third question with another yes, successfully introducing an implant-free of the version of the technique. It is the first demonstration that optogenics, which uses a combination of light and genetic engineering to control brain cells, can accurately switch the cells on and off without the need for surgery. And then he goes on to say this is the result of 16 years of research. Optogenics usually involves genetically engineering animal brains to express light-sensitive proteins called opsins. in the membranes of neurons. The opson's reactions to pulses of light can either induce a neuron to fire or suppress its ability to fire. Optogenetics has been used to map brain pathways, identify how complex behaviors are regulated, create false memories in mice, and much more. It's also been used to develop an optogenic pacemaker, among other technologies. Now let's go down right here to this. All right, so it says most optogenic techniques involve injecting viruses with an opsin gene of choice directly into the brain with a needle. To avoid this, the Stanford team used a type of PHP virus developed at Caltech that can be injected into the blood. The virus then crosses the blood-brain barrier, sound familiar, to deliver its payload, an opsin gene, to brain cells. In this case, even the delivery of the gene is non-invasive. No needle penetrates the brain. And I believe that this makes, it says the team tested the effectiveness of this on the system. In one instance, they used light to quickly and precisely stop seizures in epileptic mice, and in another, turn on serotonin-inducing neurons to promote social behavior in mice. So yeah, and I'm pretty sure what it says at the top there is that they can now engineer into the brain the ability to produce light instead of having to do it with surgery. So now they can genetically engineer that. So if you remember in certain genetic therapies that are being pushed on the public right now, gene therapies, there was the luciferase 666, and I'm sure that'll trigger the algorithms to censor me now. But what was the luciferase for? A lot of people thought it was just something that really didn't matter. Well, in my personal belief, it was the engineering of the gene in which they could have light-expressing opsins in the brain cells that would do that could power perhaps microbots like this. So we've already went over microbots that run on lasers. Did we or did we not? Well, we're about to. But first, let's go ahead and check out this. Actually, yeah, no, go ahead and read this article. You know what? Really quickly, now that I mentioned power, this is from another article. We're not going to read the whole thing. Really quickly, it says, everything about the scale of these tiny robots, which is talking about a completely different robot that's been created. Robots operate as impressive. According to their official paper, which I read, each microbot weighs less than 5 milligrams, and it's only a bit lighter than a single grain of sand. Meanwhile, the 3D printed device is only 2 millimeters long, which is huge in these small terms we're talking. Even more impressive is the fact that they are so small without battery technology being ready to shrink with them. To overcome this obstacle, the Georgia Tech team turned to a unique power alternative of vibrations. Seems like the stuff out of science fiction. The microbot is powered by a tiny piezoelectric actuator that is triggered, which actuator, if we remember, makes it move. That is triggered by anything from an acoustic speaker to an ultrasound device. So what would an acoustic speaker be? Anything from as small as your phone to as big as one of those big old ones you've seen at concerts. Vibrations from the actuator make the bot's legs move, propelling it forward. Despite its miniature size, the device can travel 4 times its length in 1 second. This is quite an impressive feat considering its lack of traditional power. Now, again, if you want to read the entire article, you can do so. Let's go ahead and read this this is the last article we're going to read before we check out the white papers, and 2 very quick videos that are fast forwarded so you can see kind of what I'm talking about. This is also from IEEE Spectrum. Talk about EEE. Microbots deliver stem cells in the body. Magnetically, and this is from 2018. Magnetically controlled micro robots gently carry cells to the hard to reach organs in the body. And just to put this in perspective for you really quick, 1 millimeter, so 1/10 of 1 millimeter is equivalent to 100 micrometers, and the symbol for that is a which we're going to encounter here in a moment. Okay, so let's get through this. This picture is micro robots designed to carry stem cells, fluorescent, which are marked as fluorescent green, they're actually green, throughout the body, are shown here on a cell culture plate. And I'll show you robots like this moving in a moment. They look kind of like, the astonishing thing about stem cells is that they can be coaxed in a laboratory into becoming nearly any kind of cell. Obviously, you guys know that. A group out of Hong Kong announced this week that they had, that they had invented a new delivery tool using tiny magnetically controlled robots. The cell-carrying machines move non-invasively through the body to target, site, and deliver their stem cell cargo. The researchers, led by Dong Sun, a professor at City University, Hong Kong, demonstrated the devices in zebrafish and mice and reported their success in the journal Science Robotics. The advance is notable because Sun, that's the guy's name, and his colleagues were able to demonstrate that the robots will work in animals. It's really uncertain how to make these tiny machines move in living organisms, says Bradley Nelson, a micro roboticist at ETH Zurich, which is in Germany, I assume, Zurich, who was not involved in the project. Several other groups, including Nelson's, have demonstrated cell-carrying microbot designs in computer simulations and test tubes. But in vivo is harder, he says, which has been done, as we've already seen, but we will continue to see. And it shows here the different cell types. This is what I was talking about. I believe it's called is it nanometers? Micrometers. And this is 25 micrometers. So to put it in perspective, 1 millimeter is 100 of these, supposedly, or I think it's 200. Could be 1,000. Let's go on with this. You can figure it out for yourself. Sun designed the robots to resemble birds, the prickly spherical seed pods that cling to clothing. He and his colleagues fabricated them using 3D laser lithography. He coated them in nickel for magnetic actuation and titanium for biocompatibility. The porous burr-shaped bots hold cells between their spike using magnetic fields. Researchers can guide the bots to a site of interest in the body. There, they may release the cells, which then proliferate and do their job in regenerating damaged tissue. And I'm going to show you another technology that was, I believe, designed and modeled after, I could be mistaken, by blood vessels, because they mention it in their white paper. And so it says did I already mention that they can release their cell? They released, no, I don't know if I did this. The pore-shaped bots held the cells between their spikes using magnetic fields. Researchers can guide the bot to sites of interest in the body. There they can release the cells, which then proliferate and do their job, regenerating damaged tissue. And you're going to see that they actually did that. Sun and his colleagues tested the mobility of the robots by injecting them in the yolks of anesthetized zebrafish embryos. They navigated them to specific points inside the yolk, as you can see here, while observing that the heart of the zebrafish continued to beat. Then they tested whether the robots could carry and release cells. They loaded them up with a vicarious type of cell called HeLa, which there's a link to. I'll check it out at some other point, and you guys can do it as well. And inject swarms of them into mice. So we injected swarms of these cell types into the mice. The robots did their job releasing the cells, which then proliferated in the mice. So they've already used this technology to proliferate a certain kind of cell. It could be a virus. It might be. I didn't really check it out. In the mice. But I do intend to do it. And he goes on to say the next step would be to load up microbots with stem cells and test their therapeutic capabilities. So let's go on really quickly, and I believe I've already read this, about how the other microbots are powered using even things that are sound-based, which could be as simple as you talking. Think about the scale of these things. How much is it really going to take? How much vibration is it really going to take? So this video is sped up. Go ahead and we're going to go ahead and watch this really quick, and then we're going to read these white papers, just a little bit of them, of course, just enough to make the points that I'm trying to show you. And then we'll see if there's any comments, and yeah, we'll respond to those. So really quick, this is Lasers Create Miniature Robots from Bubbles, headline Science, and this is from a channel on YouTube called American Chemical Society. Heavy in propaganda, but there's some interesting, very scary and disturbing type videos on their channel. Again, I would make it full screen, but it's going to lag up StreamYard too much. So go ahead and check this out with me. And it is fast forwarded to 1.25 speed. Here we go.
In factories, robots are widely used for many purposes, such as building cars, painting airplanes, and sewing clothing. The assembly of microscopic components, such as those used for tissue engineering.
Now, if you see, sorry to interrupt, if you see, that's 100 or micro meters or whatever it is. So that is actually small enough to move animal cells, which are usually around 10 So you're going to see in a few minutes on these white papers that this technology is very significant. So go ahead and watch the rest of this. I won't interrupt. And just try to keep that in mind, that is actually small enough to manipulate animal cells. This is 100 units. I'm going to show you one that's 50 units. And obviously you can imagine they can, and they actually have to be that minimum size, otherwise they can't influence the animal cells. Here we go.
And sewing clothing. The assembly of microscopic components, such as those used for tissue engineering or biomedical devices, requires much more intricate automation. Manufacturing has miniaturized to construct objects that are the same thickness as a sheet of paper. But it's hard to position such small pieces by hand. Lasers could be the solution. Now researchers reporting in ACS Applied Materials and Interfaces have used lasers to create miniature manufacturing robots from bubbles that lift, manipulate, and drop small pieces into interconnected structures. Scientists have used bubbles to move miniature parts in water, for example, to assemble 2D objects, like placing these donuts on a rod, or to rotate shapes in 3D space. However, these bubbles could not combine separate pieces and then move them as a single object. So, Niandong Jiao, Lianqing Liu, and colleagues wanted to build on their previous work with lasers to develop bubble microbots that can form inseparable shapes and control their movement. The researchers created microbubbles in water by focusing a laser underneath a small piece of resin. The bubble size was controlled by rapidly switching the laser on and off, with more time in the on position resulting in larger bubbles. Once the laser turned off, the bubbles dissolved slowly, dropping the resin in place. By shifting the laser's location, the researchers made a mobile robot. The bubble micro-robots can lift and drop parts, move single pieces to designated positions, and connect the components. The team also combined two bubble micro-robots with different functions that could simultaneously act as a rotational axis and push assembled objects. Unbreakable connections made by various types of joints allowed the bubble micro-robots to move 3- and 4-pronged gears, a snake-shaped chain, and a miniature 3D vehicle. The researchers say that the bubble micro-robots have implications for the future of microscopic manufacturing.
Unbelievable. That video is unbelievable, and you might not realize it until you start looking at some of this stuff. But the size that those were at, you've seen complex, and I'm going to mute it here while I show you, complex gears. Those are working down at the size small enough to engineer cells. So I just want you to imagine, try to imagine what something this size spinning at those speeds could accomplish. What it can accomplish is the ability to rapidly attach cells or rapidly disperse the cargoes of these micro robots. And this is my understanding after reading these white papers. And basically, something as sophisticated, this is from 2021, these papers are from several years ago. Something as sophisticated as a spinning wheel like this with interlocking pieces that is supposedly unbreakable. That is, look at that, turning geared wheels. This, which I'll explain to you in a moment through this white paper very clearly, that motion right there can be used to separate and deliver cells or cargoes of the micro robots that can be carried through the cell by something that's a little bit more archaic looking, something that's very basic looking, which is basically the equivalent to, you know, basically a pill. It's real, or a blood cell. It's really not that sophisticated. These are actually mind-blowing in their sophistication. This one, I think, can be used easily. It's being controlled with a laser to easily rearrange cellular structures in any manner they want. And of course, this is what they show you that they have, but they could have something way more sophisticated. Look at that vehicle. Only we saw earlier that one was 100 UM. I think it was in the previous article. This vehicle is as small as 50 UM or whatever that is. So that's micrometers or whatever the hell they are. And by the way, shout out to Low Pro Steve for making me aware of this. I think he probably might enjoy this when he sees it. So again, really quick, keep those scales in mind. 50 is small enough to carry a, it could even carry potentially up to 5 animal cells. That's the level of the stuff that you're looking at, is at the cellular level. And they make all this stuff with very advanced laser technology. But this is almost as small as a cell. Unbelievable. So I think it's like a thousandth of a millimeter. It's in the thousandths of millimeters. Anyways, I'm not sure how you say that. Now, again, real quick, here's a quick video, and then we're going to take a look at the white papers so that you understand more clearly what these things really can accomplish. And with this next video, I want you to understand these are obviously much bigger things, but try to imagine them on the micro scale, because that's the kind of stuff we're working with, and it's reactive materials that can elicit a highly precise response, such as gripping, even twisting and moving, or even, as you've seen in the other article, making legs move on robots. And I just wanted to show you this because it really does help you understand what this technology is. And they don't even need CPUs. If they can get CPUs that small, incredibly terrible for humanity. But they don't need them, and these things could be in people's blood around the world right now. Perhaps people who have taken part in the experimental gene therapy being pushed upon the public.
Plants and animals can quickly respond to changes in their environment. Think of a Venus flytrap snapping shut when a fly touches it. Ideally, soft robots built with flexible materials would mimic the intelligent and autonomous behavior seen in nature, combining sensing and controlled movement. However, replicating these actions in robots usually requires complex, clunky mechanics. A simpler design is needed where a single component can both sense a change in the environment and then react. Now, researchers reporting in ACS Applied Materials and Interfaces have created an intelligent material that curls under mechanical pressure and expands when stretched.
Look at that.
To make a soft material capable of autonomous movement, Zhao Zhao, Hongliu, and colleagues printed liquid metal into circuits on polymers that undergo large changes to their shapes when heated or cooled. These scientists had previously explored using liquid metals to create thin, flexible circuits. These circuits quickly heat up when an electric current is generated, either from an electrical source or from pressure applied to the circuit. When the soft circuits are stretched, the current drops, cooling the material. The researchers applied a nickel-infused liquid metal onto a soft polymer film and magnetically moved the liquid into lines to form an uninterrupted circuit. A silicone sealant coated the circuit. In response to a current, the soft material curled and changed from light pink to dark red as the temperature increased. The team used the material to develop autonomous grippers that sensed and responded to pressure.
That's at 4 times speed.
They could pick up a small round object and then drop it when the pressure was reduced. Another autonomous gripper transported a blueberry when the material was repeatedly stretched and released. The researchers say that these pressure and stretch-sensitive materials could be adapted for use in soft robots performing complex tasks or locomotion.
And essentially what you're seeing there is materials themselves that react to their environment if they're touched, and that was at 4 times speed before I speeded it up to 1.25 speed, so 1 1/4 speed. But essentially, as you can see, they already know how to make these materials that react. And basically, when they're stimulated either by heat, which can elicit one response, or with pressure, it can stimulate a response to just grabbing things. So think about at the small micro level that we just seen, where we're talking the size of animal cells. That's the kind of technology that's going to be going into these cellular robots, because once you can engineer stuff with metal at the level of making gears and basically making circuits. They could create a circuit within the human body to potentially receive signals, be powered by some of this organic power. And you're going to see in a moment they discuss powering these machines with things as amazing as glucose, which is sugar that can be found in the blood, and fruits and natural things like that. It's natural sugar. And so if they could form these circuits and they could form these spinning gears inside of you at this level, then they could really do anything they want. And this channel also has a very interesting video, the American Chemical Society. I want you all to check it out. Quite the propaganda-laden thing. Its emblem is a triangular eagle, not just an eagle, but a triangular eagle and a triangle upside down. And they have a thing about like kindergarten science, 5th grade science, and one of the very first like 2nd grade science things is atoms can be rearranged to make different chemicals and different chemical structures and stuff like that. Something like that. Look, I mean, I can't remember 2nd grade or 5th grade microbiology or whatever the hell this stuff is. But go ahead and check out all this stuff is in the description. And again, a millimeter is like 1/1000th of a micrometer or something crazy like that. You can look it up. You're all intelligent. So really quickly, we're going to go over these 3 white papers, and then that'll be it. And we'll check out if there's any comments. Controlled manipulation of multiple cells using catalytic microbots. And this is from 2010. And this is from core.ac.uk, and you can even download the PDF. So really quick, it says self-propelled microjet engines. Microbots can transport multiple cells in specific locations in a fluid. The motion is externally controlled by a magnetic field, which allows to selectively load, transport, and deliver the cells. And they're going to go on to tell you that they use hydrogen peroxide as fuel. That's right. They already have micro engines, as they describe them, or sorry, nano engines or something like that. Nano machines right here. They do reference them as engines, being small engines that can propel these vehicles. And after you read some of these white papers, they only take a few minutes. They're like 3 pages long, 4 pages long, some of them. You really get the idea of they talk about loads and fuel and nano engines and nano machines of a movie where there's truckers that volunteer to drive these trucks full of life-saving stuff into people's bodies. I think it would be a crazy movie. But anyways, let's get through this here. Self-propelled microjet, because that's really what they're doing. They're doing fleets of these at once and making them drop cargo and pick up things and whatever they want. All right, so the development of useful micro or nanomachines, which could one day be manipulated inside the human body, remains a challenging dream in nanotechnology. Yeah, right. And biomedicine. Over the last 5 years, there's been substantial interest in the use of chemistry to propel tiny engines in similar fashion that nature uses biochemistry to power biological motors. Consequently, researchers have fabricated self-propelled nanomachines capable of performing useful tasks such as the transport of synthetic cargoes. Artificial nanomachines that are able to swim along with biological matter such as cells have yet to be achieved. So that's what it's saying. Artificial nanomachines that are able to swim along with biological matter, such as cells, has yet to be achieved. But that's not true, as we've seen with the other literature from much more recent. This is from 2010. But this is kind of important for people's understanding here. We're not going to read the whole thing. Only a few more sentences, basically. Excuse me. There are 3 main challenges that researchers try to conquer when engineering artificial nanomachines. Efficient self-propulsion, demonstrated by the catalytic breakdown of hydrogen over oxygen, some. Basically, I don't understand those things. It's hydrogen breaking down into oxygen, forming bubbles, propelling the machines. Catalysts and catalyze enzyme contained in nanomotors. There it is, nanomotors. Motion control achieved by the incorporation of Ni or Fe segments and I think those are metals, and subsequently using external magnetic fields to orient the nanomotors. And the development of useful tasks such as the transport of cargo like microparticles and nanoplates in a fluid. And it will even go on to tell you that by the end of this, the conclusion is, they say to date, let me see, to date, however, the transport of single spherical micro nanoparticles has been the last result accomplished by nanomotors. On the other hand, micro jet engines, dubbed microbots, based on rolled up nanotechnology, have demonstrated the ability to transport multiple micro objects, not only colloidal microparticles, but also metallic nanoplates. Nonetheless, the transport of biological materials such as cells by artificial nanomachines has not been achieved so far. But they're going to tell you at the end of the conclusion of this that actually they did. Nonetheless, so it says they can't transport cells, but we've already seen they basically can. And they're going to tell you in a moment that they can. In this paper, here we report on the pickup transport of the release of multiple neuronal CAD cells, which is a cathochrominogenetic cell line from a cerebral nervous system. And so I looked this up, and the other definition was something basically that receives light in, what do you call it, in engineering. It's something that receives light and reacts to it, which is very interesting considering we were just seeing that many of these need light to do certain things. Perhaps there's different not actuators, perhaps there's different mechanisms on there to respond to different stimuli. Anyways, so here, let's read this on. Although large cells are loaded at the front of the end of the microbots, their motion is not totally halted. The micro engines are self-propelled by the release of oxygen bubbles generated in the captivity of the microtubes. And we're going to show you the microtubes there in a minute so you can really understand this. From the catalytic decomposition of peroxide used as fuel. See, I told you. Their motion is coordinated by an external magnetic field, which, once is turned rapidly, enables the release of the loaded cell to its desired target. Essentially, they turn it at a 90-degree angle, and it separates the cells reliably to their location, but they have to do it once more to get off all of the cellular material. So anyways, it goes on to say how they were done, and it's very technical, sputtering, silicon wafers, all that stuff. So I'm going to go ahead and read the so it says the microbots are formed by a hollow tubular structure containing a thin PT layer, which I believe is again an element in their inside. When the micron tubes are immersed in a hydrogen peroxide solution, microbubbles are generated in one or more nucleic points of the PT layer. And then this is the last sentence for this stuff. Due to the catalytic decomposition. They already told us that. I don't know why they told us. So this is, again, look at that. CAD cell attached to this so-called micro robot swarm, or whatever this is. It's a tubular micro robot, essentially. And that's what I was saying. It could be like, it kind of looks like a truck. These are one of the main ones that they use to transport cells, because the CAD cell is an animal cell. So you can go ahead and look it up for yourself. That's what it is. And basically just told us that anyways. So that is only 10, what is it called again? A micrometer, I believe it is. Could be an anyways. It's something different here, but I'm not going to go ahead and click on that. So it says in the Google search. So anyways, yeah. That is only 10 whatever those are wide. It's extremely small. I showed you one so far that was 100. Then I showed you one that was 50, and this is from years ago, 2010, and this is only 10 wide. So it's pretty incredible. And they go on to say basically that they got this idea. They didn't say they got the idea, but they said it really mimics something in nature, which is the blood cell, which can do similar things for transporting stuff when it contracts and releases. So Anyway, let's finish this off. Sophisticated micro machines, like we seen earlier, I think. Sophisticated micro machines are required for the performance of multiple tasks. For instance, the consecutive loading of several cells is beneficial for complex applications such as cell sorting. Therefore, high power output and easy loading mechanisms are demanded for this purpose. The loading of multiple CAD cells during a period from 10 to 20 seconds is shown in videos 2, 3, and 4 in ESI. That's something we didn't see. The microbots can overcome the drag force generated by the loaded cells. Although their speed is reduced, the microbots can travel at speeds of roughly 40 per second while transporting large biological material. Analysts from the videos, our experiments clearly indicate that microbots could be suitable in the near future for the development of bioanalytical and biomedical application using different kinds of cells. In summary, we have demonstrated that the catalytic microbots can transport animal cells in a controllable manner and release them to desired targets. Earlier it said, to date, we have not seen anything that can transport animal cells. Right here it says, in summary, we have demonstrated catalytic microbots can transport animal cells in a controlled manner and release them to desired targets. There you go. Told you. So that's it for this paper. You can read it yourself if you want. The links are in the description. And it says the presented work paves the way for the future of biomedical applications by using artificial nano micro machines. For instance, the microbots can be used for cell sorting or to move one single cell into an analytical device. Example, lab on a chip. To analyze changes in biochemistry, physiology, metabolism, or single cell. And it says one can see in the not distant future, microbots could transport drugs to cure unhealthy cells. And it says it goes on to say it could also separate cancer cells and blah, blah, blah. So what we've seen earlier was, in my opinion, some pretty sophisticated micro machines. And like I said, if you read all this together, it really seems like they're talking about, it really seems that this indicates, since this was 10 years ago, and we're reading something from almost 11 years later, 2021, 11 years later, I just showed you a video of basically micro machines. Listen, in the steam powered revolution and the industrial revolution, when steam engines were invented, it was simple. The simple ability to move large machinery with stimuli, an example, steam. So now that they're at these small levels, building gears and building other machines, technically machines, that can reliably manipulate cells and cellular structure, then it seems like they could right now destroy your whole cellular structure, or perhaps engineer something inside of you that wasn't there before. Really anything they want. So it's pretty interesting, and I hope you get what I'm trying to say there. One more really quick thing, and then a medium thing right here, and then we're done. Recently, a significant amount of attention has been paid towards the development of man-made synthetic catalytic micro and nano motors that can mimic biological counterparts in terms of propulsion power, motion control, and speed. However, only a few applications of such self-propelled vehicles have been described. Here, the magnetic control of self-propelled catalytic TIFEPT rolled up microtubes that I just showed you, and I showed you the tubes, yeah. I did show you them, yeah. Just trying to remember myself here. And TIFEPT refers to elements, of course. The microtubes, which are called microbots, that can be used to perform various tasks, such as selective loading, transportation, and delivery of microscale objects in a fluid is shown. For instance, it demonstrates the polystyrene particles and thin metallic films, nanoplates, microbots self-propelled by ejecting microbubbles, like we just seen, via platinum catalytic decomposition of hydrogen peroxide in oxygen and water, and the fuel and sufficient concentrations are optimized by attaining a maximum speed, are you ready for this, of 275 UMs per second. That's pretty, it's 5.5 body lengths, and they're not talking about human body lengths, they're talking about of the robot, I believe, per second at least 15% of peroxide fuel. The microbots, and we all know peroxide is safe. As far as I know, it's safe in the bloodstream. It is safe in your mouth, and it is safe. You can drink it, I believe. The microbots, fact check drinking it. Don't be drinking it unless you fact check that, but I'm pretty sure you can. You can definitely use it for your mouth. The microbots, I wouldn't drink it though. The microbots exert a force around 3.77 pn when transporting a single 5 diameter particle. And again, animal cells are 10 nanometers or whatever the heck we're talking about, that measurement. So that's half the size of a human cell, or an animal cell rather. Evidencing a high propulsion power that allows for the transport of up to 60 microparticles by the introduction of a Fe thin film into a rolled up microtubes. Their motion can be fully controlled by an external magnetic field. Now, I think this is pretty interesting. Check out this. Richard Feynman, in his 1959 speech, said, I believe this is about swallowing the surgeon, there's plenty of room. At the bottom, envision microscopic factories with tiny machines that can perform sophisticated tasks. The dream of manufacturing nanomachines is to mimic biological motors that can convert chemical into mechanical energy and hence develop diverse functions. Some fascinating examples in nature are DNA and RNA, polymerase, rotary motors such as ATP synthesis, cilia and Fenglia motors, diosins, kinesins, and myosins, catalytic synthetic micro and nano motors, and engines are of growing interest because of their high locomotive power and easy control. Several approaches have been focused on the implementation of catalytic conversion of chemicals, an example of hydrogen peroxide into kinetic energy, and control of movement over nanomotors by different sources has been reported, as we've already went over. The transport of spherical particles used as cargo was recently achieved through electrostatic, chemical, or magnetic interactions to the attached cargo. Extra functionality has been given to the cargo, magnetic properties, chemical modification for electrostatic binding for loading into nanomotors. So they've got nanomotors, and they can use these other small cellular transportation vehicles to load them into nanomotors. Very, very interesting stuff. And again, right here it says some fascinating examples of this are DNA and RNA. So essentially, in my opinion, what they're trying to do with some of this experimental gene therapy that's going on right now is they want to remove people's immune system's ability to fight disease and re-engineer it so that microbots are your immune system. So even if you are one of these humans that's in their society and they don't totally eradicate or assimilate humans within 100, 200 years, we're talking long term here, you'll still be some level of cyborg, and perhaps maybe even in a worse way. We seen earlier in the article they can influence social behavior with serotonin production. So anyways, let's go ahead and go down a little more, because there is something I want to read off here before we give it a rest. All right, so let's go down. All right, it's right down here. I believe it's page 5. So here's some of the stuff they were showing you. See, when we're talking about nano robots, one of them is a tube. When they're guiding one of them, they can essentially see what's happening to a lot of them, because of course you can see they're end to end these darn things. They're so-called nanobots. Here's some interesting things. I guess this is showing what way these things can move and how they can be maneuvered quite easily, almost like a spaceship in the sky. And yeah, one of these plates is 50 wide and 10 nm, which is a different figure, tall. Anyway, whatever that means. These plates, again, look at these plates. They could be used to build circuits. They could be used to influence machinery, like we seen earlier, micro machinery, rather. And they could be used even as receptors for an electrical charge, perhaps. I'm not sure. But I think I'm doing a pretty good job for someone who has a grade 8 education. Can you imagine I read all this stuff by myself without an educational assistant here to help me? All right. So it says synchronize the magnetic field with movements of the tubes. Therefore, with straight motion, straight motion of microbots was desired. If they desired that, the magnet was left stationary. In contrast, by rotating the magnet and thus changing the direction of the magnetic field, microbots align themselves with the magnetic field. Being able to remotely control these microbots is an essential requirement for specialized and sophisticated tasks such as wireless transport and delivery of microscale loads and building blocks in a fluid. Figure 1C envisions the concept of a microfactory where micro objects are mixed together with synthetic microbots in a fluid and fuel is added in order to power them. And I'm guessing that's a self-contained thing, so it doesn't mess with the blood. And see, again, once they get down to this level, they can build these things. They could build things inside of the blood where they could power these things, where they could accomplish things they wouldn't already be able to do, because the need for an excessive amount of chemical fuel, such as hydrogen peroxide. And then when it breaks, it's not a big deal. A bit of hydrogen peroxide at the microscale is not going to hurt you. And then they could influence you in other ways, perhaps creating a bunch of micro machines in one of these micro factories that they're talking about here. This isn't me. This is a white paper, which is an academic paper. All right. So it's not crazy. I wouldn't conspiracy theorist here. Anyways, let's go ahead and read the conclusion of this. And it talks about putting in a small, what is it? Is it an actuator? Something that, where is it? I think it's a catalytic converter, something that helps to create a magnetic thing inside there that's, it says it's a permanent magnet. Maybe it's down here anyways. But basically, it does say within this document that they were able to put a permanent magnet inside. So that way they don't even need to do this, and they can just basically have a remote control. So let's go right down to the conclusion here. It's on page 5. Maybe some of you are reading along at home as you watch this. Page 5, children. Conclusions. We have demonstrated that the directed loading and transport of micro objects by high propulsion powered rolled up microbots driven by microbubble propulsion mechanisms. The loading of particles was facilitated by the pumping of fluid into the microtubes via catalytic reaction. The ferromagnetic thin films introduced into the microtubes assisted the wireless motion directed by external magnetic fields. The extraordinary easy control over the movement of the microbots by changing the direction of the magnetic field during motion helped to accurately load and deliver the cargo desired places. Our microbots had enough power to carry great numbers of particles, or even particles without any modification. We demonstrated that tubular microbots can load and delivery with different kinds of materials, namely both polymetric and metallic particles. The presented results are encouraging and constructive universal drug delivery systems. However, biocompatible fuels such as glucose would then have to be used, and these systems would need to be explored further. And that's about it for that one. So let's just check out this last little bit of document. It said, and this is from something multimodal chemical magnetic self-propelling self-propelling microbots. And I'll read something else here in a minute. But it says, all right, so let's read this here. We demonstrated a reciprocating motion towards and away from the target. The delivery of the motor to a target of animal cells and to a fro motion inside micro channel, which remotely controlled direction and steering of the motor while it was in motion, including the ability to halt the motor, control on the directionability of the micromotor together, and the control over its speed, employing both the external and internal fields. Could make this device potentially a candidate for the next generation of self-reliant and remotely controlled micro machines. The attachment of biomolecules, drugs, and cargoes with enhanced biocapability could open up technology viability of these objects for the application in imagining sensing, transport, and drug delivery inside biological environments. And at one point, it does say they want to use these for sensing the environment, not some environment, but the environment and sensitive ecosystems without interfering But basically, at that point, they could even engineer. At this point, they can engineer almost animals. Almost, they can almost engineer animals or synthetic creatures at the molecular level. And I think something like that would be a little bit harder to destroy, depending on what you use to make it. And these could also be self-healing. I mean, anyways. So enough of that. But yeah, essentially, that's pretty much all we have. We're going to go ahead and look at some comments. I went way longer than I wanted to do, but I think a lot of this was very important stuff. Please do share with this video. I'm sure at least the first 20 minutes will be pretty interesting to even the most blue-pilled person. But yeah, I was just trying to find the thing where they said they put in an it's the same thing that's in a car. It's a anyways, the thing that generates magnetic field by itself, and you can look it up. I think it's called a catalytic converter or some crap like that. Anyways, let's go ahead and see if there's any comments. We went a little long, but again, I think that was worth it. That's one of the better videos I've done in the last month. And I worked, I wouldn't say really hard on it, but I did spend, a couple hours on it. So please do, again, go ahead and share this out with your friends. Let them know that we do this here. Of course, we smoke on the channel. We have a good time, but we also like to talk about more serious stuff like this, especially in an age where stuff like experimental gene therapies are being pushed upon people, and they're not even aware that this technology has existed for 10 years and now is being perfected to the point where they can create gears inside of people's bodies. One more time before we go, let's just look at it, where they can create stuff at the micro scale, pick up and drop stuff. They've got stuff that can move at that. Look at how, and I think that's showing how long it is. Stuff that can move like that, they can position these cells, they can make entire factories now. This is from 2021, January 20th, 2021. Look at what they can do. That little thing that's moving, that's only 100 of these units. So in the middle spaces, those are big enough to move cells and stuff. Absolutely crazy. And we've seen to remove cells earlier, like I said, they had to flick it at a 90-degree angle. I don't think they have to do that when this kind of technology has just been released. I'm sure it wasn't just created. It's absolutely incredible, though. And when you look at these gears, you can really see what kind of power they can have. Like I said, the Industrial Revolution took place because big gears turning like this, little things turning like that with power being produced. Imagine where we're going now in the 21st century with microbiology and transhumanist agenda. In my opinion, they want to replace bacteria, they want to replace your immune system, and they want to have total control over your body. And if you don't think that this will be used for just that, I think you're living in a crazy cotton candy unicorn populated fantasy world, and you just can't wake up to the facts. Hey, we got to do what we can to blue pill these people. Don't look at them as NPC or sheeple. I don't think that were always aware of what was going on. I'm sure you could think back to a time when you really believed the world was a good place and the government was on your side. You might have been 5 years old, but hey, some people wake up at 5, some people wake up at 55. I'm Al Wooten, aka the Skeptical Canuck. We're going to check with the comments now. And remember, we do all kinds of content all week, all year, all month long. We're going to be cranking it up. Let's check out the comments. We'll see if there's any. Yes, Warren Ferry, masks affect the people that wear them more than the other people. He says, and that comment's from 1:41 a.m. He says, Jesus sucks. I'm not sure why you say that. He said the diabetes and cancer epidemics are not an accident. No, I'm sure they're not. I'm sure they could have gotten rid of that stuff long ago, and they perhaps even engineered it. Who knows what they've been messing around with? And how much does a good haircut cost? Not too much. I should have washed my hair before I started this. I woke up with bedhead earlier, but I've been working on this stuff all evening. So, yeah, you're right. But no, I'm actually going to go ahead and shave my head next month, although it will look a lot better once I wash it. It always does. It's beautiful Guido hair. And yeah, so if you've got any questions, throw them in now. And yeah, like I said, in June, I'm going to be shaving that stuff. I want to go exactly 6 months with this haircut. Anyway, thank you very much for tuning in. And Yeah, remember, we're going to be pumping out all kinds of content probably this week before the end of the week. Stuff coming up, including police refusing to enforce lockdowns and the lockdowns, I think, being lifted, and some of the stay-at-home orders being abandoned here in Canada, and a bunch of other things, including drone strike in the USA. Roger Stone claims to have survived a drone strike, and if you see the video, it does not look like a gas explosion. And also some details about Revolution Possible and how a coming revolution could be something to make you think that they're crushing down the people who've been betraying us, but in reality, they're empowering something much, much worse. Thank you, everyone who tuned in, especially Warren Ferry. Have a great night, and don't forget to share this stuff. Oh, even a little thing.